Mitosis and Meiosis. First, we'll take you through the steps of mitosis. Mitosis is a process that a somatic cell divides into two daughter cells. It is an important process in the development of normal organisms. Both growth and repair make mitosis an important part of life. If mitosis were to get out of control, diseases such as cancer may occur. Interphase is the first step of the cell cycle and occurs before mitosis. In interphase, the cell prepares for mitosis by growing in size and replicating its DNA. Interphase consists of the G0 phase, G1, S, and G2 phases. Prophase is the first step of mitosis. In prophase, chromatin condenses into chromosomes. Nucleoli disappear and the nuclear membrane breaks down. Chromosomes move toward the center of the cell, and the mitotic spindle starts to appear. In prometaphase, duplicated genetic material is separated. The nuclear envelope breaks down, freeing the sister chromatids. The kinetochore forms around the centromere, and the kinetochore microtubules attach to the kinetochore. In metaphase, spindles form. Chromosomes position along the metaphase plate located on the outside of the spindle. Double-stranded chromosomes are attached to a spindle microtubule at its centromere. In anaphase, centromeres of each chromosome separate. Each sister chromatid now has its own centromere and is called a chromosome. There are twice as many chromosomes as there were in metaphase. Spindle microtubules move chromosomes. In telophase, chromosomes gather at the poles and start to form daughter nuclei. Daughter nuclei will be identical to the nucleus of the parent cell. Nuclear membrane begins to reform, nucleoli begin to reappear, and chromosomes start to disappear. Cytokinesis divides the cytoplasm. Each cell gets about half the materials and organelles in the cell. The whole process of mitosis creates two daughter cells with a diploid number of chromosomes, meaning they are completely identical to their parents. Now we'll take you through the steps of meiosis. Meiosis is a phase in sexually reproductive organisms. It is of great importance because it creates genetic diversity within the population. Meiosis I separates homologous chromosomes. In premeiotic interphase, all chromosomes are duplicated in a process similar to that of mitosis. Outside the nucleus are two centrosomes, each containing a pair of centrioles. These centrosomes are produced from one centrosome being duplicated. They serve as microtubule organizing centers. In prophase I, DNA is exchanged between homologous chromosomes by recombination, which often results in chromosomal crossover. The paired and replicated chromosomes have two chromosomes and four chromatids. The pairing of homologous chromosomes is called synapsis. Non-sister chromatids may cross over at points called chiasmata. The nuclear envelope is broken down. In metaphase I, homologous pairs move along the metaphase plate. Tetrads line up. Centrioles are at opposite poles, and microtubules from the centrioles attach to the kinetochore. In anaphase I, kinetochore microtubules shorten and pull homologous chromosomes apart. Whole chromosomes are pulled to opposite poles, forming two haploid sets. Each chromosome has a pair of sister chromatids. Non-kinetochore microtubules lengthen, pushing centrioles apart. The cell elongates to prepare for division. In telophase I, chromosomes arrive at the poles. Each daughter cell has half the number of chromosomes, but each consists of a pair of chromatids. Microtubules disappear and a new nuclear membrane surrounds each haploid set. Chromosomes uncoil back into chromatin. Cytokinesis divides cytoplasm and the membrane pinches to create two daughter cells. Meiosis II separates sister chromatids. In prophase II, centrioles duplicate. The two pairs of centrioles separate into two centromeres. The nuclear envelope breaks down and the spindle apparatus forms. In metaphase II, each daughter cell completes the formation of a spindle apparatus. Single chromosomes align along the metaphase plate. Kinetochores of sister chromatids face opposite poles. Each is attached to the kinetochore microtubule coming from that pole. In anaphase II, centromeres separate and the two chromatids of each chromosome move to opposite poles on the spindle. The separated chromatids are now called chromosomes in their own right. In telophase II, a nuclear envelope forms around each set of chromosomes. Cytokinesis takes place, dividing each of the two cells into two new cells. The final result of meiosis is four daughter cells, each with a haploid set of chromosomes, meaning they have half the number of chromosomes of the original parent cell. The crossing over in meiosis creates new genetic combinations on the chromosomes of the daughter cells. That concludes our presentation of mitosis and meiosis.